Okay, so this is the last lecture on photosynthesis that's happening online because I am not in town on Monday morning for class. So what we're going to be doing here is you got to get the PDF of the slides from Oaks. That's going to be this screen over here because the resolution is not going to be as good as what you can get when you get the PDF. And then I'll be working a whiteboard over here as I'm talking. All right, so you got to be focusing on this to get notes. All right, so we're going to be looking at three different things. We're going to be talking about completing the details for photosynthetic electron transport. We'll be talking about the Calvin-Benson cycle and the how it fixes carbon dioxide to make glyceraldehyde 3-phosphate. And we'll be talking about a couple of alternative non-cyclic electron transport pathways that just generate an additional proton gradient without ending up making an ADPH, and so you can make more ATP. Cells will do this when they need to generate additional ATP beyond what they need to consume in the Calvin-Benson cycle. All right, so that's where we're going. So to remind you of where we were last time, so this is the sequence. This slide was up on Friday. This is the sequence of electron transport, in which you remember we've got photosystem 2 here, the cytochrome B6F complex in photosystem 1. That's plastoquinone, and that's plastocyanin. So you don't need to know as much detail as you did in mitochondria, where you're having to learn all of the steps in the complexes and all the redox molecules. Here you have to sort of know this level of detail as far as what's happening. So if we sketch this out to pick up where we left off before, I'm going to get the cell in here, or the thylakoid network, and it looks like this. So this is going to be photosystem 1, right? And here's our bilayer. We're going to keep going over there. Alright, so here's photosystem 1. This is the NADP reductase. And you can see that my handwriting is just as bad as it is when I work on the board. And here's the reaction center chlorophyll. So here are the two electrons. This is all the light coming in. Let's see if I can do this without knocking over my clipboard. There are the excited electrons. They're going to get picked off by ferredoxin. That's the shuttle. Right. They go like that, and they go over here. And when you're done, you end up making NADPH. So there's the NADP going in. Right. And there's the NADPH. And the two electrons that we're talking about are now right there. Okay, so that's the point of all of that. And so you're left behind with something that's positively charged. So you now have an electron deficiency over here that you need to refill. Okay, and that's where we finished class on Friday. So how do we get there? Well, we have to go back to photosystem 2 to make that happen. So if you back out, this is the biggest part of this diagram over here, and if you back out to photosystem 2, you've got the same thing going on. So if we sort of connect this, right? There's the cytochrome complex. Get this all on one page. That would be good. Looks like that. All right. So let's remember where we are. This is the stroma if we're in a chloroplast. It would be the cytoplasm if we were in a bacteria. Here's the lumen. It's the thylakoid lumen. All right. All right. So this is the cytochrome B6F complex, right? And this is photosystem 2. Okay. So what's happening in here to fill this in is you've got the electrons that are in photosystem 2. They're absorbing all this light energy that is resonating in through resonance energy transfer, right? Which we learned earlier on Friday. These electrons, if they're in the reaction center, they're going to get super excited and they're going to get picked off by plastoquinone. So there's a shuttle right here, which is plastoquinone. Okay? So plastoquinone is the shuttle, and plastoquinone is the thing that really contributes to all of the proton gradient, because it works just like coenzyme Q, where it's going to pick up two electrons. Right? When it picks up two electrons, it'll pick them up in sequence just because it's the same quinone ring. It picks up the first electron. It'll pick up a second electron, then it'll pick up a couple of protons, right? So they're going to get bound by plastoquinone, and now you have PQH2 2e minus, It's the same binding affinity thing. It's no longer held with high affinity, so right up in here on the PowerPoint, and it's going to diffuse away. All right, so it diffuses out, and when it's in this state of PQH2, right, 2e minus, like this. Remember that it's moving in the bilayer. It's got the same long fatty acid tails on it, and the only place that it can bind when it's in its PQH2 site with high affinity is right down here. So there's a binding site right there that it will end up in, and it's on the lumen side. So it's going to look like this. All right. And so what happens? So this actually works the same way as in the, this is just like the coenzyme Q cytochrome C reductase complex. All right, so what's not depicted in this image, well, it is, there's this little ball that says Q on it. That's the alternative quinone binding site, because there's actually a second one. But what's going to happen in here was when PQH2 2e minus binds with high affinity, right? So you have the same high affinity binding, right? It's going to bind, and the protons are going to come off. And you remember the sequence of what happens here. The first electron goes through some redox molecules that you don't need to worry about, and it's going to get picked up over here by plastocyanin, right? Which is the shuttle that works between the cytochrome complex and photosystem 1, and so they're going to pick up one electron at a time, because plastocyanin is just like cytochrome C in the mitochondria. All right? And when it picks it up, it's going to go over here. It's peripherally associated. It'll bind when it's at, so here it's starting out at +3, right? And now it's at +2 because it's picked up an electron. Okay, same logic. The electrons are going to go back, and it's going to replace the electron in photosystem 1. Now the thing that's kind of cool, if you go back to the cytochrome complex and you look at what's going on in here. So what does the PowerPoint show us? The PowerPoint shows us that the protons are coming off through plastoquinone, but also there's this line of additional proton flow. Now this is really from a recycling step, right? And you don't need to know the steps that are in here, but the second electron, right, makes its way through the redox molecules that are in here, because up here you have the site of a second plastoquinone binding when it binds when it's proton and electron deficient, right? And this electron is going to get picked up like that. Then you have another plastoquinone coming in from photosystem 2 where it's PQH2 2e minus, just like you learned before. It's going to bind down here because this guy diffuses away. So a second example comes in, sends its electrons, one electron to plastocyanin, protons come off, the second electron comes over like this, right? This now has 2 electrons on it, and it'll pick up protons like that, and then it'll leave. It'll feed back in. Okay? So if that doesn't make any sense, you need to go back and you need to look at what happens with the coenzyme Q cytochrome C reductase, because it's the same principle. Right? This evolved really early in the evolution of capturing energy and electron transport in the ancestral prokaryotes, and so you have a common pathway in mitochondria and in chloroplasts. Right? But ultimately all these electrons are going in and they're refilling photosystem 1 right there. All right, so and you're left behind when you do this with this positively charged photosystem 2. And to finish that off, this is the third part in the path. And if you're looking at the third part and you're trying to figure out what's going on, there are these little dotted blue lines to tell you where things are happening. All right, and so in the third one, you're now left with a positively charged photosystem 2, and this is more straightforward. Right, this internal thing in here is the most complicated in the sequence, but the third step here, what happens is you have this positive charge, and it's going to get replenished from the water splitting reaction. So the last thing that's going to go on in here is you've got water, right, that looks like this. These are the electrons in water, and it gets split. So it gets split to protons and half of an oxygen, if you're keeping track of your stoichiometry. And these electrons are going to go in and replace the electron deficiency in photosystem 2. All right? So, and the thing that makes this work is the stuff I talked about on Friday about the redox values. The photosystem 2, when it's electron deficient, its redox value is plus 1200 millivolts, okay, which is a better electron acceptor than the water-oxygen couple, which runs at plus 816 millivolts, so that'll actually drive the splitting of water and the generation of oxygen, and then you're done. And that's how it works. So your beginning point for oxygen donating for oxygen donor. For electron donors is always coming from water going to oxygen, okay? And your ultimate electron acceptor here is NADP+, right? That's what's going on. Now, the whole point of this is the same point that you get in the mitochondria, right? So when you're looking at what's going on in here, you're making ATP, so I can't really fit this on here. So if we take this out, through the wonders of the whiteboard, get another piece of paper. And we look at how this is going to go. It's going to look something like this. All right. So this is the thylakoid membrane. All right. So here's the lumen. Here's the thylakoid membrane. All right. This was the whole electron transport chain. That is right here. All right. And you've now made this proton gradient because you're moving protons across. So you've accumulated protons. Protons are going to accumulate in the lumen. They're going to go through the ATP synthase, like this, and you're going to make ATP. So you end up going like this. Right? It's the same rotational catalysis mechanism I taught you earlier for what happens in the mitochondria. They're very similar. So you're making ATP, and remember you make ATP in the stroma. Right? That's really critical. The other thing you got to remember that you made in the stroma is NADPH. Right? And the reason you care about that is you're going to use the ATP and you're going to use the NADPH in the Calvin-Benson cycle. All right? Now, so let's go in there. So if you look at the Calvin-Benson cycle, it looks like this. So this is this terrifying cycle that looks like it has about 18 steps, and it really does. You don't have to learn all the steps. Really all you're learning is the imprint of carbon up at the top, the export of glyceraldehyde 3-phosphate down at the bottom here, all right? And you can break this into 3 functional groups. You can break it into carboxylation, which is this beginning point, when you're fixing carbon dioxide and RUBP to make phosphoglycerate. Right, you have reduction, which is here. What happens with reduction is you're reorganizing the 3-phosphoglycerate into glyceraldehyde 3-phosphate. One of these gets exported. Then you have regeneration, because you got to go back and you got to remake the ribulose 1,5-bisphosphate like this, and the regeneration step takes some more ATP, and it actually takes phosphate to make it happen. All right, so these numbers don't seem to make any sense until you look at the balance. So I'm going to play this movie that's on here. You can see the movie better, not this tiny thing when you go on Oaks and download the movie and look at it, but here it is.
So far, energy from light has been stored in the chemical bonds of ATP and NADPH. In the Calvin cycle, this stored energy is used to produce sugar molecules. The Calvin cycle is a complex series of chemical reactions carried out in the stroma. The Calvin cycle begins with carbon fixation. 3 molecules of carbon dioxide are added to 3 molecules of a 5-carbon sugar, abbreviated RUBP. These molecules then split up to form 6 3-carbon molecules. In the next 2 reactions, products from the light reactions are used to boost the energy of these 3-carbon molecules. First, high-energy phosphate groups are added. Electrons are added in the next reaction. The added electrons reduce the 3-carbon compound, creating 6 high-energy G3P molecules. G3P, or glyceraldehyde 3-phosphate, is a sugar, the final product of the Calvin cycle. One of the G3P molecules represents the 3-carbon dioxide molecules fixed so far. The other 5 G3Ps are reshuffled to regenerate the original RUBP molecules. To summarize, so far the Calvin cycle has used the energy of the light reactions to reduce 3 molecules of carbon dioxide and produce 1 molecule of G3P. 3 more carbon dioxide molecules are fixed to form G3P in this same complicated way. To make each G3P, the Calvin cycle consumes 9 ATP molecules and 6 NADPH molecules. These are regenerated in the light reactions. G3P is the actual final product of the Calvin cycle. The cell can combine two G3Ps to make glucose, which stores the energy that chlorophyll originally captured from the sun.
Okay, so this pathway we don't cover in as much depth as we covered like glycolysis and the citric acid cycle. All right. Really what you want to get to understand this is you want to organize it like this. All right, so if you think about this, you've got CO2 going in, you have ribulose 1,5 bisphosphate, all right, and ribulose 1,5 bisphosphate and carbon dioxide come together to make 3-phosphoglycerate. Okay, now, and you can't quite see the edge of that. There we go. Looks a little better. Now, the 3-phosphoglycerate is going to get reorganized into glyceraldehyde 3-phosphate. Alright, and some of the glyceraldehyde 3-phosphate gets used to remake ribulose 1,5 bisphosphate. Alright, so there's a bunch of steps here. Now, when you go around like this, not all of it gets remade into RUBP, some of it gets exported. Like that. Okay, so this looks confusing. It's confusing until you count the carbons. So if you start looking at carbons, if you figure out how many carbon molecules these are, you can make sense out of this. Alright, so CO2 is 1 carbon, right? So, okay, RUBP is a ribose. It's a ribose sugar that's modified, so it's going to be 5 carbons. So this is 5 carbons. Okay. If you combine 1 plus 5, you would get 6. 3-phosphoglycerate is 3 carbons, so you'd need 2 of these to get 6, right? Because then 1 plus 5 equals 6. So it's easy math, okay? You're going to split it in half and you're going to reorganize it. You have these 3 carbon molecules. It's going to get reorganized into a 3 carbon molecule over here, which is glyceraldehyde 3-phosphate. All right. So if you're trying to keep track of that, this doesn't really make any sense because you're left with 3 carbons over here, glyceraldehyde 3-phosphate. You're left with 3 carbons over here. It's not as simple as one of these 3-phosphoglycerates going into G3P that gets exported, the other going here, because you can't take a 3 carbon molecule and turn it back into a 5 carbon molecule. It's not going to work. All right. The other thing that's not matching up in here is if you're thinking about the carbon load, if you're going to export a 3 carbon molecule, that means you got to bring 3 carbons in, otherwise it's not going to balance, right? That's why this formula up here is 3 CO2s makes 3 ribulose 1,5 bisphosphates into 6 phosphoglycerates, because what's really got to go on in here is if you're going to make this work, you got to multiply your carbon input by 3, because you're exporting 1 G3P. So you multiply this times 3 equals 3 carbons. 3 carbons go in, 3 carbons come out. If you're going to multiply that by 3, then you're doing this reaction 3 times. You've got to multiply this by 3. So you've got to multiply ribulose 1,5 bisphosphate times 3, because this happens 3 times. And then 1 times 3 is 3, 5 times 3 is 15. Alright? So what's helpful about that is you can then add this up. 15 plus 3 should be 18. If you're going to get this to 18, you need to multiply this by 3 times. So you need to go times 3. It's going to equal 18. Alright? Now you got 18 carbons. So now you got 15 plus 3 makes 18. And what happens in here is when you set this up is you now have not just a couple of these you now have 6 copies, right? Because 2 times 3 you have 6 copies of G3PG. Right? Because 2 times 3 is 6 times 3 carbons. So you got 6 of these. So that's great. So you still got 6 of these. You reorganize them into G3P. You have 6 of these. That still equals 18. Alright? You take one out. You're exporting one of these. 3 carbons out. That means you're left with 15 carbons over here. Because 18 minus 3 is 15. So you take this. You got to get to 15. Then you need 5 of these. 5 times 3 is 15. 5 times 3 is 15. All adds up to 15. 15 plus 3 is 18. That's how it works. So you can export 1 3-carbon molecule for each 3 CO2s you bring in. That's how you balance the carbon load. All right. Now the thing to think about what the relationship of this is to photosynthetic electron transport and the light reactions is where do you use the ATP and the NADPH. All right. So when you're organizing this, all right. So this part in here, right, is really the carboxylation. All right. Okay. This part over here is really the reduction. And what's happening over the reduction is this. Reduction is going to use ATP and it's going to use NADPH. So the ATP that gets made in the light reactions is consumed in the reduction step. Okay. The PIs don't come off because the PIs end up on the glyceraldehyde 3-phosphate, the way you balance all the phosphates. All right. The other thing that happens in here is this is also where you use up the NADPH. All right. So there are 2 steps in here. I don't care if you know that, but you need to know that NADPH is also getting used up. So you go back to NADP+. Right? So these 2 molecules right here come directly from photosynthetic electron transport in the ATP synthase. All right? What you also have going on, and that's how you get to the G3P. And then when you're going to regenerate it, you got to get this 3-carbon G3P back to ribulose 1,5-bisphosphate. There's a bunch of steps in here. It's not actually linear. It's pretty complicated. But what happens in here is you use some more ATP, so you actually have ATP that gets consumed in here, and this is also coming from the ATP synthase. To turn this back around, all right, and you need to do, and the phosphate that comes off of the ATP ends up coming into here to make 1,5-bisphosphate. So it works like that. So that's the other point where you're putting in the energy, and these steps across here, right, are called the regeneration part. Like that. Okay? Now, when you go back to the chloroplast, if you look at the PowerPoint, so this is the stroma, where these reactions are going on, and the inner chloroplast membrane right here is where you have a translocator, and what this thing is doing is it's exchanging glyceraldehyde 3-phosphate for phosphate molecules, because you got to be bringing phosphate in because you're exporting something with phosphate. So you have to have this exchange, otherwise you'd run out of phosphate in the stroma. It wouldn't work. So this antiport's going on in here, and when you get to the outer membrane, it just goes right through because the outer membrane has pores in it. Remember, they can pass anything up to 12 kilodaltons, and this is well below that. It's going to come out. Now, when it gets out into the cytoplasm, it does this. This is the bottom half of this figure in the book, right? Glyceraldehyde 3-phosphate is going to get made back into sucrose in the cytoplasm. So if we get this image out of here, go back and you look at the whole chloroplast, right, it works like this. Alright, so if you take your chloroplast, right, here's your membrane, this is the thylakoid, here's the stroma, where all this is happening, right, here's the Calvin-Benson cycle. You need to know more than this. You need to know what I just taught you, right? And this is G3P. Here's the transporter. that exchanges G3P for phosphate. Tries to go in. And then the G3P can make its way out into the cytoplasm. Right. This is the glyceraldehyde 3-phosphate, and it's going to get regenerated into glucose. And on into sucrose. That's happening in the cytoplasm. Here's the stroma, right? Here's the lumen. Works like that. Now, the sucrose synthesis pathway is, a lot of it is similar to glycolysis. It's just running the other way. If you look at the intermediates in here, which are not intermediates you need to worry about, but you've seen this before, fructose 1,6-bisphosphate, fructose 1-phosphate, glucose 1-phosphate. Well, it's glucose 6-phosphate in glycolysis, but it's going like that. Yeah, so the thing in the book is, when you're looking at it, it's counting these as 6 carbons in here because it's 6 carbons per glucose, but if you're trying to make this add up, and you go back and look at your original one, remember that you've got glyceraldehyde 3-phosphate on its own as a 3-carbon molecule, and when you put it together and you end up with glucose, which is 6 carbons, if you think back to glycolysis, right, in glucose when it splits at dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, it splits into 2 3-carbon intermediates, right, so that's what's going on in the numbering. That's why when you look in here, the numbers don't seem to add up, so you've got 6 CO2s coming in, but the 6 CO2s that are going in are counted up in the 1 glucose that's coming out. And the way I'm teaching it to you is you're exporting 1 glyceraldehyde 3-phosphate per loop of this whole thing, and so that's 3 carbons, so you have 3 CO2s coming in, and then the numbers add up. It's easier to do the math. Alright, so that's the Calvin-Benson cycle, and that's the level of detail you need to know as far as what's going on with the carbohydrate chemistry inside the chloroplast. So, if we go up into here, so there's 2 other pathways which are pretty cool for trying to figure out what's happening with the electron transport chain. So chloroplasts don't just do linear electron transport. Chloroplasts can also do a cyclic electron transport that doesn't end up making a net output of NADPH. So you're not dumping all of this energy into the Calvin-Benson cycle, you're actually reusing the NADPH to run the process around again. As a result of that, you can make more of a proton gradient and make more ATP. That's what's happening right here. Right, so the only thing that's different in here is the part that's outlined in red here, right? And it's using up the NADPH that is being generated. So instead of having photosystem 2 in here, you've only got this other thing called NADPH reductase. Right, so this is the cyclic flow. And if you look at what's the same, right? You've got the cytochrome complex. You've got photosystem 1. So we'll do the same stuff in black, like this. You've got plastoquinone. You've got plasticyanine. Right. You've got electrons getting excited here. Going up here, they're getting picked up by ferredoxin. You're making NADPH. Right, so it's going like that. And you're making NADPH. Right? There are the electrons. If you look at how all that's going, plastoquinone is picking up its protons, right? Dropping them off. And it comes over here, and it's PQH2. Let's use our red proton pen, right? And the electrons are going like this. One of them's going over here. This thing you already know. There's the electrons. They're making their way over there through the shuttle. Right, so there's the other plasticyanine. As it moves across, there's only one plasticyanine. And then you've got the other recycling thing that we talked about just a little while ago. We have the other plastoquinone site. and get more protons going in, right? And this is all contributing to making ATP. Used for ATP. That's what it says down there behind the mouse pad. Alright, but the thing that's different in here is where the plastoquinone is binding. So there's another place that plastoquinone can bind. This is kind of interesting because if you think back to the mitochondria, you remember that you had complex 1, NADH goes to coenzyme Q, you had complex 2, right? Succinate goes to coenzyme Q, you had the fatty acid oxidation pathway where you end up making the FADH2 which feeds into complex 2, right? And you had the you have the recycling step in the coenzyme Q cytochrome C reductase. So you have these multiple sources of high energy plastoquinone when it's going to pick up some protons and electrons. And here you've got the same thing going on in chloroplasts. You have this thing over here that's called the NADP reductase. All right. It's actually called the NADPH reductase. What's also interesting about this is the NADPH reductase is using NADPH right here, right? Turning around and remaking NADP+. NADPH has a couple of electrons on it. These electrons feed into this pathway, and they get picked up by plastoquinone. As a consequence of this, you have proton flow that occurs. Alright? So if you go back and look at what you learned about complex 1 in the mitochondria, the NADH coenzyme Q reductase, the NADH coenzyme Q reductase takes NADH from the citric acid cycle, pulls off the electrons, generates NAD+. Those electrons make their way through the sequence of redox molecules, eventually get picked up by coenzyme Q, right? And you have the T helix, which is responsible for the additional proton flow going in here. So this is really similar to that. It turns out there's a functional similarity here, which is pretty cool. But that's the other source of electrons for plastoquinone. And the result of all this, since you have this loop of NAD, pH, NAD going around here, you're not making that to feed into the reduction step in the Calvin-Benson cycle. You're just making more of a proton gradient so you can make even more ATP. That's the advantage. So there are times when chloroplasts need to do that because not all the ATP is only used for making carbohydrate. Right? Particularly if you're a bacteria or something like that, where you have if you're a photosynthetic bacteria, that also can be your one of your sole or your main energy source. So you have to be able to make more ATP than you just need for fixing carbohydrate. Right? The other one, and this is the last thing we'll talk about, is there's a cyclic electron flow in prokaryotes which generates only ATP, and this is all you're doing. So in this guy, this is different than the cyclic flow that uses the NADPH reductase, because here all that's happening is this is looping around in here, and what's different in here is that the cytochrome shuttle, so plastocyanin, instead of going and binding to photosystem 1, it loops back around and it rebinds to this photosystem. So you have a single photosystem in here, right, which isn't really photosystem 1 or photosystem 2, it's got binding sites that work for both. Right? So if you think back to what we learned before, right, here's photosystem 1. Photosystem 1 has a binding site for plastocyanin. Photosystem 2 has a binding site for plastoquinone. All right? If you look at the prokaryotic model for nonlinear flow, the photosystem has a binding site for this cytochrome BC1 complex, which is very similar to plastocyanin. It's different, but it's very similar. And it has a binding site for plastoquinone. And it absorbs electrons, right? You're not splitting water in here. All you're doing is your electrons are looping around. So all you're doing is you're using light energy to make a proton gradient. And that proton gradient is used to make ATP. All right, so this looks more complicated than it really is, because if you dissect it, it's not that hard. So let's get our last piece of whiteboard up here. Let's think about this one. So this is the cyclic flow. So this is prokaryotic cyclic flow for only ATP, making only ATP. So the way this is going to work is you got a membrane in here, right? Alright, if we look at where we are. So where are we? So this is going to be the cytoplasm, because it's a bacteria. So pay attention to that. This is extracellular. So this is the photosystem. Right? This is the cytochrome complex. This is the ATP synthase. Right? So you only have two complexes as far as the electron transport sequence, but you got two shuttles, the same two shuttles. They're just landing, the cytochrome one just is landing in a different place. Alright? So this photosystem, when it's working like photosystem 2, what's happening here is light's getting absorbed. Right? Electrons are going to get excited. This is plastoquinone. They're going to get picked up. Since it's plastoquinone and it picks up electrons, it's also going to pick up protons. Right, like that. Now it's in its PQ H2 2E- state. It's going to bind down here because the cytochrome complex, just like all the other ones, only has a binding site that can bind PQH2 2E- with high affinity on the extracellular side of this plasma membrane. So your electrons are going to go into here. Protons are going to come across. Right? That's contributing to the proton gradient for the ATP synthase. But the interesting thing that's different on here is if you look at what happens with plastocyanin, so this is actually cytochrome BC1, right, which is similar but not exactly like plastocyanin. That's cytochrome C, right, from the mitochondria. It's going to pick up an electron. When it picks up an electron, it's going to change from its +3 to its +2 state. And it's going to leave, and it's peripherally associated with the outer surface of the plasma membrane, and the only place it can bind is over here. So it's going to land over here as cytochrome BC1, and when it lands, its electrons are going to go back and replace the electron lost from the photosystem. Alright? Now, if you complete your idea about what's happening in here, remember that this cytochrome complex has a second plastoquinone binding site, just like all the other ones do, right? And the second electron from plastoquinone is going to come up here. It's going to land there. You're going to get another plastoquinone binding. You get more proton flow, right? And that's going to contribute to the loop in here, so it gets all the energy out. And you're going to have multiple copies of cytochrome BC1 going back and forth, because you got to have a second one coming over here to deliver the second electron. Just like that. And that's the loop. Now all you're doing, and this is really efficient, all you're doing is you're taking light energy, right? You're using the light energy to make ATP. So if you're a prokaryote and you're in the cytoplasm and you need ATP, there it is. You've used light energy, you've directly captured light energy to run this proton gradient, and then the proton gradient is consumed to make ATP in the cytoplasm, just like that. So there's a lot of variations. So that's the end of what you need to get out of the photosynthesis stuff. And what we'll do on Wednesday is we'll start working on transcription and translation, so we go into a new area. And if you haven't gotten it yet, you need to get the images, you need to get the PowerPoint here off of Oaks. I'm also going to take still photos of these whiteboard things and stick those in a file so you can get those as well, in case you can't read the chicken scratch on the animation as it's going along. But I would really encourage you to look at that as a last resort and to really be taking the notes, treating this like a real lecture and taking the notes on your own. If you just passively read all this stuff, it's not going to work as well. Because this is one of the lectures, so it's going to be on the second exam, and there's 10 lectures going into exam 2, and this is one of them. So there'll be 10 points worth of material coming out of the stuff I talked about here. Alright, so you guys have a good day, and I'll see you on Wednesday.